Greetings to all my viewers. Myself, Dr. Raktim Mukherjee, Associate Professor and Head of the Department, Shri P.M. Patel Institute of PG Studies and Research in Science, affiliated to Sardar Patel University, Anand. My current series of videos will deal with the broad topic, the cells and organs of the immune system. In the very first video, I will lead you to the formation of immune cells. But to exactly form the immune cells, we require a process called as hematopoiesis. So let's have an overview of hematopoiesis in this particular video. For immune response, we always require immune cells. In this connection, the hematopoietic stem cells or the process of hematopoiesis is very important. Now, when you talk about immune response, the immune response results from the coordinated activity of several types of cells and organs. To delve deeper into the topic, I have divided into a series of videos. First, I will deal with hematopoiesis. Thereafter, I will deal with cells of the innate immune system, followed by cells of the adaptive immune system. Thereafter, I would deal with various organs of the immune system, that's the primary lymphoid organs and the secondary lymphoid organs, together with the microenvironment that are present in the body, which also leads to the immune response. Now, when you talk about hematopoietic stem cells, let's break the word and study. When you talk about hematopoiesis, now, if you understand the word hematopoiesis, this is made up of two words. Hem means blood and poiesis means formation. In very simple words, the term hematopoiesis means formation of new blood cells. So before the formation of new blood cells take place, we have to understand what is hematopoietic stem cells, because it is said that all immune cells arise from this hematopoietic stem cell. So what is this hematopoietic stem cell? Hematopoietic stem cell, abbreviated as HSCs, these HSCs have the ability to differentiate into many types of blood cells. In fact, all the red and white blood cells, that is RBCs and WBCs, develop from this pluripotent hematopoietic stem cells during the process which I described earlier, hematopoiesis. Hem means blood and poiesis means formation. Thus, hematopoiesis is highly regulated process. It's a very regulated process. This process makes a remarkable capacity to differentiate into various types of blood cells, including RBCs, the WBCs, the platelets, this capacity makes them the pivotal players of the body's continuous production of blood cells. This is a process called as hematopoiesis. Now, in adult vertebrates, hematopoiesis occurs in the bone marrow. Now here I would like to add a disclaimer before I say it occurs in bone marrow. Try to understand, at least when I discuss about humans, it need not be the case that the embryo develops into bone marrow from the very outset. It is only after the organogenesis that the bones are formed. So do you mean to say that the blood cells are not formed before the bone marrow? No, that is not the case. I will take two window period. First window period I will take is of 3 months. One is 3 months and other will be 3 weeks. During the first 3 weeks, the site of hematopoiesis is yolk sac, whereas by the time a human attains the age of 3 months, the hematopoiesis starts in liver. It's only after that the hematopoiesis occurs in the bone marrow as given in this slide. So of course, majority of the hematopoietic stem cells are continuously renewed and directed to differentiate into two major progenitor cells. These two major progenitor cells are common myeloid progenitor cells, which we will abbreviate as CMP, and a common lymphoid progenitor cells, which we shall abbreviate as CLP. So during this course of my talk, I will brief you about these two types of myeloid and lymphoid progenitor cells and what are the products that these progenitor cells produces, which in our subject shall be the cells of the immune system. Further, I would like to add that hematopoiesis is highly regulated process. It's highly regulated. And it occurs in bone marrow and, as I described, occasionally in other tissues during the embryonic development. The process is tightly controlled by interplay of various growth factors. I will abbreviate it as GF, growth factors, cytokines, the various signaling pathways. These all factors control or govern the process of hematopoiesis. Now, I am not dealing with the entire control process because that's not the subject of my today's talk. Ours is cells and organs of the immune system. So we'll be basically talking about the blood cells that become formed as a result of the hematopoiesis. Now when you talk about the cells of the immune system, this slide I have adapted from Professor Dr. Colin Hewitt's lecture. So these are the various types of the cells of the immune system. You have the macrophage and monocyte system, the neutrophils, especially the polymorphonuclear neutrophils, the eosinophil, the basophil, and the mast cell. You talk about the monocyte and macrophage, they take part in phagocytosis and antigen presentation. The neutrophils, again phagocytosis and its antibacterial in action. Eosinophil is antiparasite, and eosinophil count also increases during hypersensitivity or your allergic response. Basophil, it protects the mucosal surfaces. There is a role of basophil even in hypersensitivity or allergy. Mast cells has so many preformed mediators. These mediators will be released during mast cell degranulation or hypersensitivity type 1. Mast cells also protect the mucosal surfaces, and they are the major players along with basophils during allergic response. Now try to understand, students, all these cells that I described, they all arise from the pluripotent hematopoietic stem cell. The pluripotent hematopoietic stem cell has myeloid progenitor, and all these cells, the macrophage, monocyte, the neutrophil, the eosinophil, the basophil and the mast cells, they all originate from this myeloid progenitor. Apart from myeloid progenitor, you also have a common lymphoid progenitor, which is abbreviated, as we saw earlier, CLP, common lymphoid progenitor. These common lymphoid progenitors will give rise to the lymphocytes. Again, we know lymphocytes are component of adaptive immunity. We basically have two types of lymphocytes, that's the T lymphocytes and the B lymphocytes. Now it's time to look at the cells in the human blood. Whenever we talk about the cells in human blood, they differ in number as well as the lifespan. So let's get an overview for the same. Now, what are the features of cells in human blood? Of course, we won't look at the entire table. I would like to highlight some of the facts. Someone asks you an MCQ, which is the most abundant cell in the human blood? Your answer would be the neutrophil. Neutrophil will be most abundant. It's most abundant. Most abundant is neutrophil, 50 to 70%. You talk about monocytes, 2 to 12%. Eosinophil is very less, 1 to 3%, but in cases of certain common cold or certain types of infections, the count rises. You call it as eosinophilia. Basophil is again less than 1%. And mast cell is again less than 1%. But of course, during allergic response, the mast cell titer would increase. Lymphocytes, the total adaptive cells of the immune system, total you have 20 to 40%, out of which the T lymphocyte is 7 to 24% and the B lymphocytes are 1 to 10%. Mind you, students, it is the B lymphocyte which is the humoral arm of the immune system, produces antibodies. T lymphocytes, you have two subtypes, T helper and T cytotoxic, the two broad half type. So in other words, the total leukocyte that is present is always 7.3 into 10 raised to 3. The genesis time for RBC, the lifespan of RBC is 120 days. So whether you donate blood or you do not donate blood, your RBCs are definitely going to die after 120 days. Platelets 5 to 10 days. You know, platelet transfusion is done during some of the diseases such as dengue, where the platelet count drops. Neutrophils 6 hours to 2 days. Monocyte days to months. Eosinophil 5 to 12 days. Basophil hours to days. Mast cell again hours to days, and lymphocytes several days to years. So these are where the lifespan of the various cells in the human blood. We continue further. When you talk about the four main types of cells that develop from the myeloid progenitor, this was the hematopoietic stem cell. You remember, this is a self-renewing. It means it maintains the number of hematopoietic stem cell. Hematopoietic stem cell has two major lineages, a myeloid progenitor and a lymphoid progenitor. You talk about the myeloid progenitor, the common myeloid progenitor, it gives rise to four major types of cells. The RBCs, that's your erythrocytes. The RBCs or the erythrocytes, there it is. The monocytes, the granulocytes. When you talk about the granulocyte means those cells, those blood cells which contain granules. So accordingly they are divided into neutrophil, eosinophil or basophil depending on the biochemical nature of granules, whether it is acidic granule or basic granules. Basic granule, as the name suggests, basophil. Further, the mast cell also develops from the common myeloid progenitor. Okay, mast cell has several granules. Now what we would do, we would like to see each of these cells in details. So which are the four main common myeloid progenitor? You have the red cells, erythrocytes, the monocytes. When you talk about monocytes, coming to monocytes, monocytes migrate into tissues, and after they migrate into tissues, they would differentiate into macrophages. These macrophages would help in repair, remodel, as well as for destruction of pathogens. And the macrophages also present antigen when need arises. These macrophages can also differentiate into dendritic cells. Dendritic cells have high degree of function as they function as ingesters of antigen. That is, dendritic cells play a role in antigen presentation, a process by which a large antigen is broken down into smaller peptides and thereafter it is processed and presented in the context of MHC to naive T lymphocytes for initial activation. This can be a T helper cell or it can be for T cytotoxic cell for its activation. Coming to the third subtype, when you talk about the monocytes when they migrate to the tissues, they become macrophages. Now depending on which tissue the monocyte migrates, you have different types of macrophages. We'll have some of the macrophages in various location and function. Liver, the macrophage is called as Kupffer cells. Lung, alveolar cells. Spleen, its marginal zone macrophage and red pulp macrophage. Bone, the macrophages are osteoclast. Now we will discuss the cells from common myeloid progenitors, that is CMP. The four main types of cells that develop from common myeloid progenitors, you have third one, that is granulocyte. Amongst that you have the first, that is neutrophil. They cause direct harm to the pathogen. Basophil or mast cell is responsible for inflammation and allergic response. Eosinophil has antiviral activity and antiparasite activity. Next in the list is megakaryocytes. That's the fourth from the common myeloid progenitor. This is the diagrammatic overview of megakaryocytes. Megakaryocytes are large multinucleated cells from common myeloid progenitor or the CMP. When you talk about megakaryocytes, one of the role is platelet production. The platelets play a very crucial role in blood clotting and wound healing. There is a process called as endomitosis. What is endomitosis? The megakaryocytes undergo endomitosis. Endomitosis suggests they replicate the DNA multiple times without cell division to generate platelet precursor. These platelet precursor are called as the proplatelets. I'll write down. The platelet precursors are called as proplatelets. These are platelet precursors by the process endomitosis. Further, platelets are vital for hemostasis. What is hemostasis? Hem is blood, stasis is cessation. So you need to stop your blood flow so that the blood simply should not ooze out, and once the blood flow is stopped, only then the wound healing and the clot formation takes place. So platelet is very vital for hemostasis. Further, the megakaryocyte production is regulated by thrombopoietin. Thrombopoietin, abbreviated as TPO. Any abnormality in the regulation of megakaryocytes will lead to several disorders such as low platelet count or high platelet count. Now we come to individual granulocytes. Neutrophil, it causes direct harm to the pathogen. Basophil or mast cells, inflammation, allergy. Eosinophil, antiviral activity. The types of cells develop from common lymphoid progenitors. Of course, here you have B cell, T cell and natural killer cells, the three subtypes, and innate lymphoid cells, that is ILCs. Since majority of them are components of adaptive immune system, in this video I won't describe them in detail. So finally, I thank you for the attention. In this lecture, we talked about hematopoiesis and the formation of various blood cells. In the next video, I will take you to the cells of the immune system, where we will talk about the cells of the innate immune system. Till then, thank you very much.